biophysical investigation of DNA binding interactions of push-pull and implications for vitro anti-cancer activity, and many more. So now I would like to invite P. Ravindra Sir, Department of Computer Science, onto the dais. Now I request our today's speaker to speak and continue the session.
Okay, thanks for the invitation. Can you all hear me? You can all hear me, right? Okay, thank you, ma'am. Thank you for the nice introduction, and thank you, Mr. Ravindra, for having me here to give this lecture. And I will try to make this as informal as possible, and I will try to speak as loud as I can. If at any point of time, if you're not able to hear me, please raise your hand. And at any point of my lecture, if you have a question, just raise your hand. Don't hesitate to ask me any questions. Okay. Whatever little I know, I'll try to pass on that knowledge to you guys. So that's you've heard my name, and I work at K.B.N campus. That's the main building of our campus. It's a very lush green campus. It's one of the most beautiful campuses, one of the top 10 beautiful campuses in India. We work somewhere near the main building, not exactly in the main building, but the adjacent building. And that's my email address. If anybody has any question, you can drop me an email. That's my page here. Next slide. Our lab is interested in understanding biological systems. When I say biological systems, it can be bacteria, virus, animals, plants, humans, everybody. Yeah? And we use different tools, for example, theoretical chemistry or artificial intelligence or mathematics or computer science to understand these biological systems. Okay? So I'll give you a flavor of what we do in the lab a little later. But what I'm going to show you now is that in the next slide I have my outline. Yeah. So since Ravindra sir told me that okay, this is mostly for students, so I thought I'll introduce you to the scientific methodology. Like, we all say we are scientists or profs or whatever it is, and then we do science in the lab. So I'll just give you an example of how scientists think, and also make you feel that you are also scientists. Okay? We'll do that exercise, yeah? And then I'll talk about how we use artificial intelligence to understand the biological systems. And also I'll give you an example of how we are using the buzzword these days is artificial intelligence. Everybody talks about AI, right? Nobody wants to use natural intelligence. Everybody wants to use artificial intelligence. So we also use that in drug discovery. And finally I'll conclude with technologies. Next slide, please. So what is a scientific method? There is a process to this entire method. Okay. The first thing is you make an observation. You see something around you, and from that observation you formulate a hypothesis. The hypothesis is basically an educated guess that can be tested. And then once you test the hypothesis, you get some results, right? You carry out some experiments and you get the results. And that result may support your hypothesis or may not support your hypothesis. And then it's an iterative process. So we will do an example to actually make you feel how scientific methodology actually works in action. Next slide, please. My apologies for bringing a US example, but it can be applied to anything, like for example, curdling of milk that people may do at home, right? So here in this case, this boy has been observing his granny make with bread, okay, for over a long period of time, and then what he asked is what makes the bread to rise. And the granny explains that it's because of the consumption of sugar by the yeast that releases a gas. That's what is responsible for the size of the bread. Is that clear? So you add sugar, you add yeast. The yeast is consuming the sugar and releasing a gas that's making the bread to become puffy. Yeah. Next slide. So then John, being a very curious kid, just like you, he wonders, what if you increase the amount of sugar? Is it going to increase the size of the bread? A reasonable guess, isn't it? Next slide, please. So he makes a hypothesis. He has been observing what's happening, and he now makes a hypothesis that if more sugar is added, then the bread will rise higher. As I said, hypothesis is an educated guess which can be tested. So if you add more sugar, he feels that the size of the bread is going to be bigger. Next slide, please. So as I said, the hypothesis is an educated guess that can be tested. Next slide, please. So this is how the experimental design is going to be. So we have a title. The title is the effect of the amount of sugar on the size of the bread. Simple experiment. The effect of the amount of sugar, that is an independent variable because that's the one that you are varying in your experiments, and the dependent variable is the size of the bread. The size of the bread depends on the amount of sugar. That's the hypothesis. So that becomes the dependent variable, and the independent variable is the amount of sugar you add. So that's the title, and the hypothesis is if the amount of sugar increases, the size of the bread also should increase. That's the hypothesis. You guys are with me, or all of you had your breakfast? Sir is like, what do you mean? It's already 10:30. I had my breakfast at 6 a.m. Right? Is sir? You body today, sir. Okay. And it's also going to finish it as the informal. So the independent variable is the sugar. Now, whenever you do these experiments, you have to do them in duplicates or triplicates, meaning like, you have to reproduce the results. You have to make them in multiplex, depending on the feasibility. And you have to keep everything constant except the amount of the sugar, right? Because that's the hypothesis you are trying to test. So everything has to be constant. What I mean by everything is the flour, where you buy the flour from, the company of the flour, the temperature of the oven, the baking temperature, and the God you believe in while you're baking the bread. You cannot change the gods. And Hindus are fortunate to have too many gods, right? So you cannot change the God during the experiment. So you have to keep everything constant and do the experiment. Next slide, please. Okay, we have done this experiment. Here are the results. You need to have a control group to make sure that the experiment is working. The control group that John has decided to use is the amount of sugar that his granny has been using for over the years. So that is 50 grams of sugar. At 50 grams of sugar, he has done the experiment. He has done the experiment 3 times. He has done the experiment in triplicate. And the average value is what we are taking as the size of the bread. Fair enough? So now you have 60 cubic centimeters as the size of the bread when you use 50 grams of sugar. If you have 25 grams, it's going to be less. When you go to 100 grams, it's less. 150, it's even lesser. Remember the hypothesis. If more sugar is added, the size of the bread should increase. But it isn't the case. That's not what is happening here, right? So that means hypothesis is wrong. Now, as I said, scientific method is an iterative process. So you go and revise your hypothesis, or you go and revise your experimental setup. So now what John has done, next slide please. Next slide. Instead of incrementing it in 50 grams of sugar, he has tried to increment the sugar by 10 grams. So we still have the control group, that is the 50 grams of sugar that Granny has been using, and you increment the amount of sugar by 10 grams. So you have 60 grams, 70 grams, 80 grams, and 90 grams. And see what happens now. The size of the bread keeps on increasing up to 70 grams of sugar. After that, it keeps going down. So now, from this experiment, what we have learned is Granny has been using The grandmother has been using 50 grams of sugar all these years. Instead, if she has used 70 grams of sugar, she would have got a bigger bread. So the optimum size amount of sugar is 70 grams. So this is a new knowledge that you have learned. So whenever you learn new knowledge by doing these experiments, you try to share that knowledge with your peers, meaning your friends, your colleagues, and you publish that data. Yeah, that's what actually happens in a scientific method. Are you guys clear about what happens in science? What experiments we do? So let's go to the next slide. Next slide. Skip this, whatever I said is there on the screen. So the major take-home points, when you say it's a scientific method, you make an observation, question, and you try to explore that particular observation further by forming and testing the hypothesis. And whatever new information you get, you are going to evaluate that information and share it with the community, scientific community. Next slide, please. Okay, now that you guys are clear about what a scientific experiment is, I'm going to show you another simple experiment that we biologists do. You guys, computer science people are very nice people, very fair people. I always respect computer science people, but biologists will see what they do. Here is a go back. Yeah, this type of. Next one. Next one. Next one. So here is the hypothesis that there are male songbirds, and these male songbirds sing because of the sex hormones that are produced by the testes of the male songbirds. So that's the hypothesis, and you need to test this hypothesis. So what do? How do you test this hypothesis? In the case of baking, we increase the amount of sugar and then we did an experiment. Here also you have to do some experiment, right? So now the next one, please. The experimental group. That's why I said biologists are very, very cruel people, right? The one standing in front of you. Right? So you have male birds with testis removed at birth. Because that's the hypothesis. You said that okay, sex hormones that are produced by the testis are responsible for the same behavior. So you just remove the testis. And then see that they grow. And you observe that okay, what happens when they grow older? Are they going to sing or not? So far so good. But when you get the final results, so when you get the final results, you will be confused to know the results. The reason is you have removed the testis. To remove the testis, you have to make an incision. You have to cut somewhere. Right? So whatever result you see, you have to make sure that it is not because of cutting that skin there, rather it is because of the removal of the testis. So now your control group will be the same, a group of male birds where you make the incision but you don't remove the testis. So you make the same surgical procedure but you don't remove the testis. Here the independent variable is presence or absence of the testis. Next one please. Dependent variable is singing period. Next one please. So male songbirds, what is the data telling you? After you allow the birds to grow, it is observed that the male songbirds without testis do not exhibit singing behavior. So is your hypothesis supported or not? You guys had your breakfast or not? Yes. Is the hypothesis supported or not? Thank you, sir. So that's my appetizer for the talk. So that's what scientists do. So we actually make some observations, we try to come up with some hypothesis and then make experiments to actually see that whether our hypothesis is supported or not. And then revise the entire process. Are there any questions at this point of time? Okay, the next topic, I'm going to show you an example of how to use machine learning in understanding biomolecular interactions. So when we look at biological systems, the first thing that comes to your mind is that, oh, these people come from BPC because they could not get MPC seat in +1, +2, right? So they're good for nothing people. Biologists are good for nothing people. But biologists, the one thing with biology is the most complicated subject to learn. It's no more about remembering the scientific name of king cobra. Nobody wants to know what is the scientific name of king cobra. But what people want to know is how do you overcome the pandemics that you get now and then, like COVID-19 pandemic. All of a sudden, the entire world started understanding about viruses. Till that time, nobody cared about viruses, right? All of a sudden, oh, there is a new variant, there is a Omicron, there is Delta, so colleges will be closed. I hope there will be new variants, many more variants, because online exams are easier than offline exams. Because offline exams, your invigilator is standing right in front of you, right? You can't copy much. I always say that you have to come up with newer copying mechanisms. Yeah, maybe have some specs or something, and then longer vision or something. So now, all of a sudden, everybody started trying understanding the biology. Biological systems are very, very simple. There is a certain information flow that happens in biological systems. Those of you who are not interested in listening to me, take a nap. I don't mind. Yeah. But it's better to reduce the noise in the background. So what happens in biological systems is there is an information that goes from one place to the other place. So in every nucleus of a cell, in the sanctum sanctorum, like the temples you see, sits the DNA. DNA carries the information to make RNA molecules, and that RNA molecule makes protein molecules. That's how the system, that's how the information transfer happens. Let me give you an example. Let's say somebody walks into a bar and then starts taking an alcoholic beverage. Okay? So the signal goes to the brain, and then it goes to the cell, it goes to the DNA. There is a part of the DNA that has information to make an enzyme called alcohol dehydrogenase. Yeah? So then the DNA gets transcribed to make the RNA. It's another RNA molecule that also has some letters in it that has the information to make alcohol dehydrogenase. And then that RNA gets converted into protein, that is the alcohol dehydrogenase. Now, the ethyl alcohol that you have taken from your beer or any other alcoholic beverage goes and binds to this alcohol dehydrogenase. And that ethyl alcohol gets converted into something else. That's why people say that drink slowly, don't drink fast, right? I know that none of you guys do it, but so you might have heard of stories where people may have said that somebody has taken illegal liquor and has gone blind, right? That is because illegal liquor has methanol instead of ethanol. So this methanol goes and binds to alcohol dehydrogenase and converts methanol to formaldehyde. And formaldehyde is very bad. That can make you go blind. So I am not propagating that you have to drink only pure liquor. No. I'm just telling you an example of how information flow happens in biological systems. DNA makes the RNA makes the proteins. So whenever we look at these systems, it's always interesting to learn how these biological systems interact with each other. Next one, please. So you have, in many cases, whenever there is a communication, in many cases, RNA molecules interact with protein molecules. We wanted to study, can we predict, if given an RNA sequence, given a protein sequence, can we predict whether they are going to interact or not? Can we tell people that, okay, given this RNA, given this protein, these guys are going to interact? So that is the challenge we had. The reason why I call it as a challenge is because in the next slide I'll show you that, the next one. There are a lot of experimental tools that are available to study these interactions, but they're very, very expensive and they take a lot of time. So people want to know, can we predict? Can computer science people help us? So that's where we chip in. Yeah. Now in the next slide, I'll show you what is the problem at hand. We are going to use machine learning. Machine learning is a very simple thing. Everybody says, machine learning, artificial intelligence, deep learning, and all that stuff. So let me explain to you what machine learning is by using this picture. What do you see in this picture? Madam. I'm calling you only. Huh? Oranges and apples. Yeah, sit down, sit down, sit down. Lemons. Alright, so we have some bunch of fruits, and there is a reason why you are able to recognize it. Right? You are able to recognize them as oranges, or you are recognizing something as apples because your parents have told you that this is what it is. As a kid, you learn, right? Parents are the ones who teach you first, right? Now we have to tell the machine to recognize them. Machines don't have brains. Somehow, I have to teach the machine to differentiate between oranges and apples. Okay? Then I can say that, okay, in this particular image, when you look at oranges, you have a very rough surface, right? Whereas apples have a smoother surface. So that I can code, I can actually inform and I can tell the machine that whenever you see an image with a rough surface, you can identify that as an orange. The other one is an apple. Okay? So in machine learning, so what we need is the data first, and then the second thing is we need the features. We have to give some features to the machine that can differentiate between oranges versus the apples. That's the simplest way I can put machine learning to you guys. It's not complicated. So we are going to use machine learning to actually predict whether a given RNA is going to interact with given protein or not. Next slide, please. So what do we have in RNA? Being from a non-biology background, the way we look at biological system is simple letters. RNA is made of 4 letters. It's a 4-letter alphabet: A, G, C, and Q. Whereas proteins have 20 amino acids, so it's made of a 20-letter alphabet. We don't care about what these letters mean because that's not important for us, right? When you give a sequence, you give this alphabet of 4 letters as RNA, alphabet of 20 letters as proteins. So these two guys interact, and the next one, please. And then they make that complex. And knowing that they interact is a very important thing in developing drugs or any kind of things. And then also, if you notice, here in the orange is the protein, green is the RNA molecule, and then the green molecule binds to the orange molecule at 2 points, 2 different locations, right? So this is one binding pocket, this is one binding pocket. Yeah? Next slide, please. So we have collected the data, like there is a lot of data of the structures, experimentally determined structures, and we extract the sequences. So we know that this sequence and this sequence, they interact with each other. Yeah? We know that because they are done experimentally. So we have collected around 953 complexes. Sir, is it okay if I move this chairs? No, that's fine. That's fine. So we have collected this data. We had a total of 953 complexes, and we had to do some data science to curate these data sets. Okay, without going into too many details, we have some. Finally, we have some 2,435 RNA protein complexes. Meaning, I have an RNA and protein sequence, a pair of sequences that I know that they interact, of ribosomal origin and non-ribosomal origin. I am not going to get into the details. If anybody is interested, I will be here till evening, so you can talk to me later. So we have the data now. We have to generate the features. Remember what we did in the apple and oranges case, right? So now we have to generate the features for these guys. So in the next slide, I will show you the kind of features we have generated. So this is the first time in the entire world, this is the first time a group has used data-driven parameters, domain knowledge, to predict biological interactions. Next slide, please. So we asked the question, we have 20 letters of the protein, 4 letters of the RNA molecules. So of these 20 letters, which letter is most frequently occurring at the binding pocket? Remember, like there are two places where the binding is happening.
At those two places, what is the letter that occurs more frequently? So that letter happens to be R. And the next most frequent is K. And we ask the same question about RNA. And it so happens that all these four letters in RNA have the same frequency. Next slide please. And then we have also asked the question, what is the length of the binding pocket, the binding interface? Is it made of 3 letters, or is it made of 5 letters, or is it made of 10 letters? And what we found is, more often than not, it is made of 5 letters in both the cases of RNA as well as proteins. Yeah? So now we have classified these letters into different groups. Next slide please. We have classified those amino acids into different groups based on the frequency of their occurrence at the binding interface. This is how we generate the features. So you have these letters, right? This is the protein, so it's a 20-letter alphabet. This is an RNA molecule, so it's a 4-letter alphabet. And then this fellow, we have classified these 20 letters into 4 different classes. And then we are using 5-mer representation. This is a K.B.N representation in machine learning terms. So now class 0, this is class 0, class 1, class 2, class 3. There are 4 possibilities, right, for any letter. It can fall into any of the 4 classes. And then we are taking 5 letters at a time. So if you look at the number of possible combinations, it will be 1024, right? The same happens with the RNA molecule. So we read, once again I'm telling you, though it may be looking complicated on the slide, simply put, you have this string of letters, we read 5 letters at a time. We make a word. Each word is made of 5 letters. And we read these words one at a time on an overlapping basis. Yeah? Next slide, please. So we have the features, we have all the data, and then we plug it into our XGBoost. Everybody uses RF or SVM. So we wanted to use boosting algorithms because boosting algorithms have their own advantages compared to the binning algorithms. So we used, for the first time, we have used XGBoost, and then within 500 iterations, we have actually optimized the entire learning process. Next slide, please. And this is what probably is familiar to you guys, the confusion matrix. Here I am showing you the accuracy, precision, recall. Accuracy is defined as true positives plus true negatives over the entire data set. And precision is true positives over true positives plus false positives. Recall value is true positives over true positives plus false negatives. When I say true positives, what I mean by that is I know that this is a positive, this is an apple. It's a true positive. And true negative is the opposite. It's a binary classification basically, yes or no. Given a pair of RNA and protein, are they interacting or not? Yes or no. I know that the pair interacts, and my algorithm predicts it as it interacts. That's a true positive. It doesn't interact, my algorithm predicts it as doesn't interact. So it becomes true negative. The other 2 cases are the false positives and false negatives. So we do a pretty good job. I mean, actually you can see that true negatives are indeed recognized as true 96% of the time. And there's a case with the true positives, 93% of the time we actually predict true positives with 93% accuracy. Next slide, please. So now we have our model. We have 2 different data sets. These are the baseline data sets that everybody uses now. And now whenever you make these models, the first thing that you have to do is you have to compare it with the state of the art. Somebody else may have done this work before you, right? It's not that you may be the first person on this planet to do it, but you may be doing it in a very different style. Remember that example that we have seen, Granny's example? Granny has been using 50 grams for how many years? For a very long time. And the boy discovers that 70 grams is the optimum. So somebody has already been doing it, and these are the people who have already been doing it, these three groups, and see how well we do compared to those groups. So these are accuracies. So the best of these three methods is around 0.9, and ours is 96.96. So we outperform all the existing methods, right? Next slide, please. And then we have tested these models on human samples, mouse samples, and then compared it with how the other models do. And there also we show that we outperform the existing models. Next slide, please. And when we have to predict the RNA protein interactions, we were able to convince the world that we can predict the one-to-one systems, the blue. So here is an RNA molecule that interacts with multiple protein molecules. We could predict almost all the interactions. Here is a protein interacting with different RNA molecules. So it's one-to-many kind of a network. We were able to predict all of them, most of them. And these are many-to-many networks. So multiple proteins interacting with multiple RNA molecules. That also we were able to predict with a very quite good confidence levels. So that's the story about how we use machine learning to understand biological systems. Now, another big problem with biological systems is that the data sizes. Data sizes are very small, and then the calculations take very long time. And to overcome that, to actually overcome those problems, we have developed MP3 vector representation. Remember that feature generation? When I talked about feature generation, like how we actually take the data and try to compute certain things from the data to generate features. So you may not always have enough data to generate the features. So you have to have a universal feature representation for proteins. So I'm not going to get into too many details. This is the first time where we have proposed the multi-purpose protein prediction vector. That's the MP3 vec. And what we do is we take the protein and then we extract all possible features and give it to you. Then you can use whatever feature you want instead of you computing the features. So that lessens a lot of work for you. And since we use a large data set and then compute the features, these features can be transferred. So here we club artificial neural networks with transfer learning. Next one, please. Any questions on that part? I think I have started around like 10:20, so I'll probably take another 15-20 minutes max. Yeah. So guys are already getting restless. Any questions? Okay, thank you. Well, let's look at how we use artificial intelligence in drug discovery. Now, everybody knows about drugs. You don't get them here, is it? I'm from Goa. Whenever I say drugs, I'm talking about medicines. The common term, drugs are of two kinds. One is the one that actually gives you hallucinations, which is supposedly bad and which is supposedly is banned actually, like you're not supposed to take these substances. So they lead to hallucinations. The other ones are medicines which lead to alleviation of pain or inflammation. I'm going to talk about those drugs. Yeah. Next one, please. Why do you need drugs? The reason. Next one, please. So we are constantly having more and more diseases, more and more infections, more and more, for example, hospital-generated infections. Like, let us say you don't have anything, you just have cough, you go to the hospital, and then you may pick up infections from the hospital itself. And most of the bugs that you pick up from hospital, like Staph aureus or Salmonella, these are multi-drug resistant. None of the drugs work. The reason is these guys have been living in the ghettos. They are living in the hospital where there is access to all kinds of medicines. So they keep on eating those medicines and become resistant. Yeah, it's like the story. I don't know how many of you have seen the movie The Princess Bride. If you haven't seen, watch it. It's a very old movie, but a very nice romantic movie there, where the protagonist, the heroine, let's call it, right? He starts taking the snake poison. For a period of time, he becomes resistant to poison. Okay, because you get used to it. Just like that, these bugs get used to these medicines and then they become resistant. So you have to you have to come up with new ways, new drugs to overcome these bugs. When I say bugs, the bacteria, viruses. So right now the hot topic is using artificial intelligence in drug discovery. I will show you why it is very important. Next slide, please. So all of you probably know the line, right? So one of my favorite movies of Rajini is Baba. How many of you have seen Baba? Now, in Baba there is a fight scene. In the movie Baba there is a fight scene where he releases his shoe. The shoe goes and hits whoever is the other guy, and the shoe comes back and then perfectly fits back into the foot of Rajnikant. Now, there are only two people who can actually make you convinced that that can work. One is Rajni and the other is Bollywood. So if anyone else does it, it doesn't look good. I mean, you won't be convinced because it's anti-physics, right? I mean, like it's against the laws of physics. Now, apparently, after that particular fight scene, Rajni gets hurt in the foot. He has to take the medicine. Now, most of the medicines we take are oral medicines. You take the pill to the mouth, right? It is the oral medicines. Very rarely you take IV. Because IV is when it's very, very urgent. Most of the times it's oral. So let's see what happens to a drug when you take it orally. And now remember, but in this case, Rajni has to take it orally and the drug has to go to his foot. That's where the infection is. Next slide, please. So you take the drug orally and then it has to go to the site of action, which is foot. Next slide, please. The first thing that has to happen for the drug is it has to dissolve. The next thing is when you are going through, imagine yourself as the drug molecule, yeah? The small molecule. When you are going through, in the mouth you have a different pH. In the stomach it's very acidic. So you are going through different pH ranges. So as you go through different pH ranges, this small molecule may change its shape or structure. So you have to worry about it. You have to predict how the molecule will change its structure as it passes through all these different areas in the body. Next slide, please. And it has to survive the intestinal bacteria. We'll keep going. Next slide, please. So there are a lot of things that this small fellow has to endure when it goes into the body. So now, artificial intelligence is being very actively being used in predicting what happens to this small molecule as it travels through the entire system. I'll show you some of the examples, some of our own work that we do in using AI in drug discovery. Next slide, please. So let me walk you through what it takes to develop a drug. Okay. If anybody is interested in setting up a pharmaceutical business, please do pay attention, because after oil industry, pharmaceutical industry is the next richest industry. It's the most profitable industry, right? Because
they can make blockbuster drugs. Now, how do you go about developing a drug? The first thing is you identify the disease that you want to work on. None of the companies will work on dengue virus because dengue is actually predominantly present in poor countries like India and Nepal, Bangladesh. Most of the pharmaceutical companies will focus only on those diseases that they can make money out of, like cancer, hypertension, COVID. They work on make some project decisions, and then you have to sacrifice some animals. You have to test them in animals, and then you get some permissions from Food and Drug Administration, and then you do some clinical trials. And once the clinical trials are done, then you can actually get the final approval. So the entire process takes around 15 years, 12 to 15 years. And the cost of making one drug molecule, getting one drug molecule into the market is anywhere between $3 billion to $5 billion. Now you start appreciating pharmaceutical companies, right? Why they actually put that high cost to the drug, because they have to get their money back. The first blockbuster drug is Viagra. How many of you have heard Viagra? Yes or no? Don't worry about sir sitting in the back. You won't. It's fine. None of you heard Viagra? Okay, so let me take a small tangent and tell you the story of Viagra. This is a very interesting story of Pfizer. So Pfizer was developing a drug against hypertension. Yeah? So they started the clinical trials. When they were having the clinical trials, what they have seen is the patients who were taking the Viagra had longer erections. Then the entire team of Pfizer came together, and then they have decided that, okay, now instead of using this medicine for hypertension, let's use it for erectile dysfunction. And then they started making billions of dollars. That's the first blockbuster drug that made $1 billion within a year of its of the drug coming into the market. So these are different procedures, and then it's a very long process. So computer science people, especially you guys, have a lot of scope to get into this domain. I'll show you how computer science helps in drug discovery. Next slide, please. So at different points in the drug discovery process, you can intervene. You can actually develop algorithms to make models to speed up the process. For example, nowadays you don't have to do animal testing. People have in silico animals, virtual animals basically. So whatever you have to do in the laboratory, you can do it in the computer. And whatever assays that we were talking about here, high throughput assays, you can do it in the computer. Okay? You can develop algorithms to do all this in the computers itself. As a matter of fact, now we have an entire diabetes human patients cohort, virtual cohort. They're not humans. They're not even like robots or something. It's a virtual human being in the computer. So whenever you have to do these clinical trials, you can actually do them in these virtual humans. That will reduce your costs. That will reduce your time that you spend on clinical trials. Yeah? That's where the computer science people come into in drug discovery. Next one, please. So I'll quickly go through the next 10 minutes. I'll try to wrap up the next 10 minutes. So we worked on COVID. COVID, everybody knows what it does. Next slide, please. So we selected a target, and then we, instead of doing the experiments, we thought, being from a computational biology background, why don't we design new drug molecules that can target SARS coronavirus 2? So we decided to target this particular protein. I'm not getting into the details, which I did yesterday for the chemistry people, but you guys are from CS, so I'll focus on the CS aspects. So here is the protein that we wanted to target. Next one, please. And then we have a bunch of small molecules. So we have programs which you can also do at home. These are
free softwares, there are some commercial packages, there are some open source. So you have open source available wherein you can actually take this target, you have this bunch of molecules, you ask the question whether this molecule will go and bind to the target or not. Next slide please. Next slide please. And what happens is, out of this 22,000 molecules that we tested, we found around 30 molecules that indeed bind to the target. So we reduce the time it takes to do experiments, right? That's where the use of computers come into drug discovery. Now I will show you a video of how these molecules go and sit in the pocket and then they stay in the pocket. This is a simulation, again using high performance computing clusters. We have done this simulation where we show how this fellow actually gets it. Okay, it's not coming up on the screen. Alright, while they are trying to fix it, I'll keep talking so that we can go through the session. So we have this 22,000 molecules, we have screened these 22,000 molecules and asked the question, okay, are they going to bind to my target or not? The target is a very critical part of the virus life cycle. So if we can actually block the target, then the virus is dead. So we took these 22,000 molecules and then asked the question, and then we found some 30 molecules that bind. Out of these 30 molecules, we have done these simulations and found that there are 3 molecules that are actually very highly potential drug candidates. So video out like that. So basically what the video shows is, you see these contact points I have shown you in the distances, right? That there is 4.4 angstroms between one atom and the other atom. These are the target amino acids, and this is the molecule. So for example, this fellow, this carbonyl oxygen, is 4.4 angstroms away from this atom. Yeah? And then we simulate, and then over a period of 100 nanoseconds, we do see that contact being there. It doesn't fall apart. So this 100 nanosecond simulation takes around 12 to 18 hours in our HPC. So if you do that in laptop, your laptop will crash. The docking you can do, the simulations you cannot do in your laptops. Next slide please. So as I said, we have zeroed in on these 3 molecules, and we have a collaboration with UK and Sweden groups. They are testing these molecules. They are making these molecules and testing whether they will work or not. So that's how you do this. You use this computer science knowledge to speed up your drug discovery process. But at the end of the day, it all comes down to this small molecule. It depends on what the properties of these small molecules are. Next slide, please. The small molecule space, the chemical space is huge. You can have billions and billions and billions of molecules. And which one will work? Nobody knows. Nobody knows. That's why it's a very long process. So you have to shorten that time period. How you can actually use AI to help shorten that number of molecules. So that's exactly what we do in the lab. Next slide, please. What we do is we try to predict the different properties of the molecules. And beforehand, we can tell you what the molecule's properties are, whether it is going to be drug-like or not. In the sense, should you waste time exploring this molecule or you shouldn't? Yeah? Next slide, please. So the motivation is we want to develop computationally efficient models. The computational parsimony comes into effect here. We want to have better molecular representations, and we also want to make sure that all these models have better performance. Next slide, please. So this is how a molecule is represented. Again, for us, we don't deal with the chemistry that much. But for us, whenever you give a molecule, we see that as like bunch of atoms. Okay, from a pure CS perspective. So here is a molecule This can be represented in different ways, and these are atom symbols. This is something called atomic number. You have some bond order, like there are some single bonds, double bonds, triple bonds. That's what the chemist tells you. So that's the domain knowledge. So we use that domain knowledge. We have action next please. We have developed a very novel way of representing these molecules to the machine. Machine is not going to look at what's on the left side. It cannot see what's on the left side, right? You have to give it in a different format. And this is the format we have developed. This is called the bond order matrix. And this is there is a one-to-one correlation. So far, what people use is that they do convert the molecules into matrices, distance matrix or distancing matrix, but there is no one-to-one correlation. So with bond order matrix, we make sure that a given molecule will have a very unique matrix associated with it. Like this. So this is our message passing neural network model. So what we do here is you take this molecule and convert it into a bond order matrix. We use what is called semi-master nodes. What these mean is that, like, instead of giving the entire molecule to the master node directly, we take the molecule and then we say that, okay, there are different functional groups within a molecule, right? We know that there is a carboxylic acid group, or there is a benzene ring, or there is a metal group. So we connect these semi-master nodes, and these semi-master nodes get connected to the master node. So what happens is when you are doing this K.B.N networks, the nodes actually carry the information, so you will know exactly what feature of the molecule is contributing to what property. Next slide, please. We'll skip this one. We'll skip this one next. So now let's look at again what we did with XRPI. We compared it with state of the art. We are doing the same thing here, whatever is existing. So if you look at the state of the art, these are the numbers. I'm actually showing you the average error ratio, and ours, the lower the number, the better it is. By the way, the lower the number, the better the performance of the model is. So here is our model with K.B.N plus semi-master node plus the bond order matrix. Now, the earlier models are developed by Google Brain. Google Brain has a huge team of engineers working for them, right? So we can't compete with Google Brain, but we are fortunate to have few wonderful, excellent undergrad students who actually have done this work. It took us like around 4 years to beat Google Brain. It's not easy to beat them because they have army of people working for them. Next slide. And not only the overall performance, we can actually show you wherever I have told it, the numbers. Our model actually predicts those properties better than the existing methods. When I say existing methods, the state of the art. Yeah. Next one, please. Okay, so that's how we use AI to understand the molecules, to do these docking studies, and to understand biological systems. Now, whatever I have shown you so far, it's all done in silico environment, meaning in computers. May not necessarily be in a laptop, but it's always maybe in an HPC, but it's always using computers only. But we gain a lot of knowledge in that biological systems. Yeah? So that's how we are using AI, developing new tools to understand biological systems. Any questions at this point of time? I usually don't do much work. After 5, I am off to the beach, not working in the lab. All the work is done by my students. I have a very good set of students. The entire credit for what I have discussed with you goes to my students only. So I have Vibhu's work I didn't discuss with you guys. Vibhu actually worked on dengue virus. So we tried to develop novel drugs to target dengue virus. He is now at AIMS New Delhi, and he passed out with his PhD already. And Satish is doing PhD with me, and he also works on flavivirus. I didn't talk about that work. Aman Akansha recently joined here into the drug design part of the lab. So the work I showed you, the first work, RNA protein interactions work, was done by a couple of wonderful undergrad students, Sanket Gupte and Karmskan Jain. Dharm is now, both of them joined for PE Honors Computer Science at Pennsylvania Goa campus. Sanket is doing his PhD at Stanford now, and Dharm is in Poland. And the other work I showed you is done by Shreyas. Shreyas and then Brunmai, there are two other guys involved in that work. And Shreyas is going to Harvard for his internship now, and others have moved on to different places. And we don't know everything, right? I mean, you will never know everything in life. So we have to collaborate with people who know better than us. So we collaborate with Professor Ashwin Srinivasan and Professor Snehanshu Shah from Computer Science Department. And whatever molecules that I showed you that we have designed, somebody has to synthesize them. So we collaborate with an organic chemist in the chemistry department. He synthesizes the molecule. So he is currently synthesizing some of the molecules we designed for treating diabetes patients. So that work I didn't talk about, but we also have that work. And then, so when you have these molecules, somebody has to test them in the animals. So we collaborate with Arnab and Indranee in Biological Science Department. We have an animal house. So we actually kill the mice using these medicines. And Vegetation is a close friend of mine who is in HPSU now, San Diego State University. He does some of the quantum mechanical calculations whenever we need some of those. And all of this is not possible without money, right? You need money to fund your students. I have to pay my students, their scholarship, or fellowship, and all that stuff. So the funding comes from, and also our computers, like, we have to buy computers also. So the funding comes from our own institute. They provide the HPC, the High Performance Computing Facility. And Department of Biotechnology, Government of India has given us grants to do this kind of work. And recently we got around $300 million from data.org. It's a collective project across the 3 campuses where we are also involved in it, where they want to actually enhance the human resources in data science and AI. Next slide, please. This is because I work in the domain of dengue virus, flavivirus infections. And flavivirus infections are caused by mosquito bite. Thank you all for listening to me very patiently. I'll be happy to answer any questions. Thank you, guys. Any questions?
Anybody? Any questions? Anyone అర్థం కాదు ఏం లేదు. ఓకే.
తెలుగులో మాట్లాడతాను సార్. తెలుగులో మాట్లాడతాను.
Thank you, Professor Ravi Prasad garu, for your valuable lecture. And I appreciate the students also. ఎందుకంటే మీలో చాలా మందికి మెషిన్ లెర్నింగ్ గురించి కొంత అవగాహన ఉంది. అలాగే కొంతమందికి మెషిన్ లెర్నింగ్ గురించి అవగాహన తెచ్చుకునే స్టేజ్లో ఉన్నారు. సో మనకి తెలియని పార్ట్ ఏంటంటే బయాలజీ కదా. సో ఈవెన్ దో మీరు ఇచ్చిన అటెన్షన్ కోసం అప్రిషియేట్ యూ. అండ్ డిఎన్ఏ ప్రోటీన్ సింథసైజేషన్ ఎలా జరుగుతుంది, ఆ ఏసీజీటీ అంటే ఏంటి, ఇవన్నీ కూడా మనకు తెలియనప్పటికీ కూడా ఎనీబడి ఇన్ ద గ్రూప్ మీకు కనుక రీసెర్చ్ మీద కనుక ఇంట్రెస్ట్ ఉంటే, సో ఐ థింక్ యూ నో దట్ కంప్యూటర్ సైన్స్ ఎక్కడెక్కడ ఎట్లా యూజ్ చేయొచ్చు. సో కంప్యూటర్ సైన్స్ మీరు బయాలజీలో ఎలా అప్లై చేసుకోవచ్చు, కెమిస్ట్రీలో ఎలా అప్లై చేసుకోవచ్చు, ఇలాంటివి. ఒకవేళ ఇఫ్ యూ వాంట్ గో యాజ్ ఎ డెవలపర్, సో గేమ్ డిజైనర్గా, వెబ్ డిజైనర్గా ఉంటే మీకు ఇది అంతగా కాకపోవచ్చు. కానీ ఇఫ్ యూ వాంట్ ఏ గుడ్ రీసెర్చ్, సో మీరు కంప్యూటర్ సైన్స్లో రీసెర్చ్ చేయాలనుకుంటే, సో కంప్యూటర్ సైన్స్ మాత్రమే కాదు, కంప్యూటర్ సైన్స్తో కలుపుకొని వెళ్ళాల్సిన విభాగాలు చాలా ఉంటాయి. సో ఇలా మనం బాటనీ కానీ, జువాలజీ కానీ, కెమిస్ట్రీ కానీ, ఈ కంప్యూటర్ సైన్స్ని ఎక్కడెక్కడ అప్లై చేయొచ్చు అనే దాని గురించి కొంత మీకు అవగాహన వచ్చిందని అనుకుంటున్నాను. అనుకోవచ్చా? సో థ్యాంక్స్ టు ప్రొఫెసర్.
learning enthusiasts ఉంటే ఆండ్రూ ఎన్జీ ఇస్ ది మాస్టర్ ఇన్ మెషిన్ లెర్నింగ్. ఆండ్రూ ఎన్జీ గారి క్లాసెస్ ఉన్నాయి కోర్సెరా, అది ఫ్రీ. అని 30 లెక్చర్స్ ఎన్నో ఉన్నాయి. మీరు నాకు తెలుసు చాలామంది మొబైల్లో ఏవేవో చూస్తూ టైం వేస్ట్ చేసుకుంటారు. రాదర్ ఇఫ్ యూ స్పెండ్ దట్ టైం ఆన్ వాచింగ్ దీస్ వీడియోస్, he does a very good job of explaining what is in there, and he actually does a good job of actually testing how much you have learned throughout that 30 lecture period. అది 1 GB or 2 GB ఉంటుంది మొత్తం డేటా అంతా కూడా మీకు ఫ్రీగా ఉంటుంది. కోర్స్లో ఇది డౌన్లోడ్ చేసుకోండి. దట్స్ అ వెరీ గుడ్ స్టార్టింగ్ పాయింట్ ఇన్ లర్నింగ్ మిషన్. ఓకే, ఆల్ ద బెస్ట్. థ్యాంక్స్.
మీరు ఫస్ట్ BCA సెక్షన్ 2 వాళ్ళు ఎవరి క్లాస్కి వాళ్ళు వెళ్ళిపోండి. ఐ థింక్ దిస్ అవర్ ఇస్ జనరల్ తెలుగు అండ్ జనరల్ ఇంగ్లీష్.